Joining us today at our webinar on aquaculture genomics. This is the last webinar of our series on genome editing and future advances, and you will hear from AQUA-FAANG and AQUA-IMPACT projects, some of the results that linked to the topic. So as FAP, we are organizing this webinar or facilitating it. And yes. And maybe maybe for the people who don't know already or who are not familiar with FAP, I just wanted to share who we are. We are the European Forum of Farm Animal Breeders. So our members are the breeding companies working on different species, as you can see from the slide, in Europe, but not limited to that. So what we are doing is we are trying to ensure the representation of our members' interests at EU level. And we are working on European policy and legislation regarding animal production and things related to animal breeding. We have a code of responsible and balanced breeding, which is called Code FABAR, and our members are applying this code. We try to engage and promote dialogue around sustainable animal breeding and farming among stakeholders. And we are also knowledge provider in different EU projects, which you can see from here, covering different species. And we also hold the Secretariat of Farm Animal Breeding and Reproduction Technology Platform, which is composed of FAP members and research institutes and academia working on the topic. And we try to develop research and innovation agendas and set priorities. We're trying to, I think, priority is to connect knowledge institutes and the private sector. And we try to promote and support research and innovation in animal breeding. That's all from our side. So some house rules. Please mute your microphones when you're not talking. If you have any questions to the speakers, or please write them to the question or chat box. Or you can also, if you want to talk, you can just raise your hand when the time comes. And this webinar will be recorded and shared on our website and social media. So if you don't want to be seen, please just close your videos. If there's any technical issues that you're facing, write it to the chat box or send an email to my colleague Maria Ramirez at fap.info. Regarding today's program, well, we will start with Antti from AQUA-IMPACT project on future advances of using genomics in breeding programs, which will be followed by Diego from AQUA-FAANG project, genome editing for precision breeding. And we have Sigrid Bradley as an invited speaker on social acceptance of NGTs, which we believe is an important topic when talking about genome editing. And then we will have feedback and feedback from our invited stakeholders, Alan Tinch from CAT and Ashe Norris from MOVI. So just the last announcement, we will, since you're interested in AQUA-FAANG and AQUA-IMPACT webinars, maybe you would be interested in our final conference of AQUA-FAANG, which is going to take place in between 11th to 13th October this year in Edinburgh. So if you would like to join us, because we will have an industry-facing day, you can always save the date first, and then you can always check our website for more information about registration. And if you would like to stay in touch with us, there are some links here, or you can just scan the QR code for AQUA-FAANG or visit our website. And for AQUA-IMPACT, you can also look at the project website or follow from Twitter. So now I give the floor to Antti. Floor is yours.
Okay, thank you. And it's a pleasure to be here today. Are you starting to see the screen? Are you able to see my screen at the moment?
Yes.
Okay, I will still put the pointer option on. So my name is Antti Kause. I'm from Finland. I work at the Natural Resources Institute Finland, which is a governmental research institute. We have a research group of about 30 people who work with animal breeding programs and developing genomic breeding value evaluations. And we work with diverse species. So we work with livestock, cattle, pigs, aquaculture, and then also with plants. And I show this because one of our main areas that we work is software which is used for breeding value evaluation, and it's commercially used around the world in many countries. And this is both an advertisement and then a disclaimer that this is one of our main focus areas. This slide I always show just to make sure that it's clear that with animal breeding, we aim to improve food production, and the targets are that the food production would be more profitable, so it makes sense to do it, and it would be more ethical and then more environmentally friendly. So these are the let's say the big goals that we have, and then it's about the techniques that how do we do it. If we look at the current status of the aquaculture breeding programs in Europe, we have 4 main farmed fish species: Atlantic salmon, rainbow trout, sea bass, and sea bream. And currently, for all these 4 species, we have the technological advances, meaning the DNA markers and computational expertise and methods to perform genomic selection in these species. Next, I will tell what it is. And by showing this graph, it doesn't mean that the traditional pedigree-based breeding would be forgotten. It will be always very useful. But the most advanced breeding programs, they use genomic selection. And this helps to breed novel traits like different diseases, product quality, fillet percentages. And genomic selection means that we replace the traditional Siodam offspring pedigree using DNA markers that are covering the whole genome. So typically we have thousands or tens of thousands of what we call SNP DNA markers. And we calculate the true genomic relationships between the animals. And we use this to understand the genetic determination of the traits and which fish are the genetically superior ones. And the logic is that when we have tens of thousands of markers that cover the whole genome, and we assume that each marker is linked to genes that have small effects on the trait of interest. So the assumption is a polygenetic model where we have many small genes with small effects on the trait. And this is now an important assumption here. And what is the benefit and the power of this approach is that in commercial breeding, we can apply this sort of breeding value evaluation model in multi-trait models. So we can have 20, 30 traits in simultaneously. We can make easily automated computational routines that takes the DNA data, takes pedigree if that exists, takes phenotypes, and all the manual work with the data handling or to understand the genetic characteristics of the traits can be minimized. And we can easily run breeding value evaluations which have millions of individuals that can be genotyped or non-genotyped. And this is like the commercial breeding value evaluation approach, the way we want to have it. And this approach has been highly successful in livestock, plants, and aquaculture, and it's like the industry routine. And next I present 4 topics which are like future advancements into which direction things are developing. These are my personal opinions, and you can challenge these also. And the first one is that because the genomic evaluation requires genotypic data, then the genotyping itself has become and will become an industry itself. So it's a business sector in itself in aquaculture species. And because it's so important, there can be a trend where aquaculture companies or aquaculture breeding companies, they can integrate the genotyping into their own activities, part of their own business profile, just like feed production has been integrated into some companies. And the reason is that then you don't need to outsource the genotyping. And what is important here is that the genotyping service itself, it's an elemental part to make better genomic tools for breeding programs. So this interaction is very important. The second topic important for future is to understand the mode of inheritance of the traits. So in the current genomic breeding value evaluation, we assume that these DNA markers each have a very small effect on the trait of interest. And the effects are normally distributed. This is the polygenetic nature of inheritance, meaning that hundreds or thousands or tens of thousands of genes impact the trait. And perhaps 90% of the traits are like this: body weight, FCR, feed conversion ratio, fillet percentage. And the current genomic evaluation models are doing fine with these sort of traits. But in aquaculture species, we find that surprisingly often the traits are impacted by genomic regions that have a large effect on the trait itself. We call them QTLs. And I have divided the QTLs into two different categories here. One is what I call dynamic QTLs, meaning that they are regions that in one of the data sets what we have, you may find it, but when you take another one, even from the same population, then this QTL disappears. And this is fairly common in our current data sets. And it means that these sort of regions are not very stable. And another class is what I call stable QTLs, meaning that these regions show permanent and consistent effects in different generations of the breeding program, or in different subpopulations of the breeding program, or even across populations globally. And traits that are behaving like this is the IPN resistant, maturity age in salmonids, fillet color, some disease resistant traits. And what is now important is that all this information is available in our datasets. And it has two consequences. One is that if we need to modify in the future our genomic evaluation models, so the statistical models, to adapt to these differences in the mode of inheritance. And the second one is that we can actually take a benefit out of this situation. And if we look at the polygenetic, fully polygenetic traits, then the genomic selection works very fine. If we find these dynamic QTLs that come and go, then actually the genomic selection can on the fly capture these and make benefit out of it. And if the effects are small, and same for the small QTLs, if they are stable, the current models can account for these very well. And now the issues arise when these QTLs have a huge effect on the traits, which we sometimes observe. And then we need to take into account these in the genomic evaluations. And also only when we validate the QTLs, we are able to make these sort of marker-assisted selection programs where we really know and have validated that these markers work perfectly across population and generations, that it's not just some sort of artifacts. And similarly, if we want to make from a large number of DNA markers a subset of markers that may be cheaper, then it's very valuable that we know that these marker effects on the traits are validated, stable, and in that way the new smaller panels that we can use are working firmly in our breeding program. All right. Then what does this mean for the breeding value evaluations? The first one is that in the future genomic breeding value evaluations, we need to have these multi-trait models that that, or it will be useful that automatically account for the different forms of inheritance of the traits, meaning that when we have the input data, then the software works in two phases. One is that it actually outputs the SNP effects on the trait, and it uses them in the genomic breeding value evaluation simultaneously. And one approach for this one is what we call SNP weighing, so that we estimate the effect of each DNA marker on the phenotype, and we use this effect as a weighing factor in the breeding value evaluation. And these models actually exist already, but they are not at the level that we can make proper multi-trait models and that they would be on solid basis. Some of the Bayesian models that are developed, they work with one trait models typically well, but in breeding programs, we typically evaluate 30, 35 or more traits simultaneously. Okay, another advancement, what is happening is that in the breeding programs where we have high or moderate number of DNA markers used, we can also generate DNA panels that can be used for a lower cost and for a larger number of fish. And when we have this breeding program with a lot of information about the markers, it's actually a very good possibility to generate informative SNP panels rather than making them randomly. And these sort of approaches are actually used already in breeding programs. Another one is that in some breeding programs, like in our breeding program, for us, it's easy to make the physical pedigree through ID tagging of the individuals. And then on the top of this, add genotyping. And this generates a data structure where we have non-genotyped and genotyped individuals. And these can be integrated into a single breeding value evaluation, which is called single step evaluation. Then we have a huge number of individuals where the selection is very efficient. One important advancement is that because we have these multiple types of marker panels, it's possible to make new innovative use of fish groups that previously have not been used in breeding programs. So we can, for example, go to slaughterhouses and take tissue samples of different types of fish that are there, when we know these fish have actually a relationship to the breeding program. So we can use all kind of novel ways to record phenotypes. Something about added value things. In AQUA-FAANG, one topic has been to study structural variants, and there's potential that there is some hidden genetic variation there that can be applied in breeding programs. This is very interesting. And then I have listed 3 things which are not so attractive, and adding more DNA markers into a breeding program, it has been proven that it's not so useful per se, because we already have a lot of markers, and it's more important what you do with the markers rather than how you model the traits. Another one is that it's possible to add gene expression of in the individuals into the breeding value evaluations. Also, this has not been so effective, and this has been a topic of work in livestock, pigs and cattle. And the second one is the interaction between genes. Also in these massive breeding value evaluations with polygenetic traits, it has not been proven very useful. Perhaps if you take one gene editing, then it may be important. Then the final slide about gene editing. I'm not going into details, but the issue here is that with the marker-assisted selection or genomic selection, if we have a favorable allele of a QTL and we want to fix it so we get the maximum benefit out of it, it's possible that it takes 5, 6, 7 generations to fix it through selection. And the logic with gene editing is that you can in one generation edit all the parents, and you get the same improvement just in one generation rather than in 5 or 10 generations. And that's the power of gene editing. And at the same time, you can simultaneously breed for all the other traits using the traditional methods. And of course, the issues is that to do gene editing, we need to actually know the actual genes. And then it's about the sustainability, consumer perception, the topics of this seminar. And then the conclusions. So my main message is that the most useful future advances come from genotyping services that evolve, and they produce more informative markers for breeding programs. And secondly, that the way we model even the current DNA marker data sets and how we combine all the information together.